Hi everyone. Today we're going to finish up and talk about triglycerides. Triglycerides are actually a four-part molecule. It is glycerol and then three fatty acids. So we're going to be talking about glycerol and fatty acids today. So let's get started. First is glycerol, and glycerol is a very simple molecule. It has three carbons and then three OH groups or hydroxyls here on the side. Of course, carbon has to have its four bonds, so we've got the hydrogens that then surround it. So here's our carbon, and then here's the 1, 2, 3, and then fourth bond. Again, these right here are our hydroxyl, and the whole thing is considered an alcohol. On the right-hand side, we see another form of glycerol. We have the OH group that's here, OH group here, and here. And then every point, so there's a point there, there's a point there, and there's a point there, represents a carbon. And so what you could actually see, it's not written here, it's the skeleton form, so it's implied, is that off of this carbon is another hydrogen. And then, of course, another hydrogen, and that would be true for the other two as well. So it's just the skeletal form of the same thing, glycerol. The other molecule is a fatty acid, obviously much more complex, but when you break it down, it's actually still pretty simple. The first part that we need to mention is the head portion of the fatty acid. So there's one head portion, and then here is another. These are two different fatty acids. The head portion is the carboxyl group, and then the back side after the carboxyl group is the tail. And the same thing is true here. This is called an unsaturated fat. This is the carboxyl group, and then this is the tail. Now the tail is actually rather simple. It is just filled with carbons which are surrounded by hydrogens. So here's a carbon, and to fulfill the bond rule, we've got the carboxyl group here. That's one bond. Let me change that pointer. This is one bond that's here. That's the second. There's the third, and there's the fourth. So carbon with two hydrogens around it. Here's another carbon with two hydrogens. Carbon, two hydrogens. Carbon, two hydrogens. All the way until we get to the last carbon within our tail, which has three carbons, because again, it still needs to have four bonds. This first one that's here is called a saturated fatty acid, and the reason it's called a saturated fatty acid is because it is saturated, like saturated as a sponge, saturated with water. It's saturated with hydrogens, so it's filled with hydrogens. The second one here is an unsaturated fat, and it is called unsaturated because it is not saturated with hydrogens. And to get away with this, since we have less hydrogens here than we do up here, we have what's called a double bond between a pair of carbons. And that double bond can appear anywhere in any number. So whether you have one double bond or 100 double bonds, it is considered an unsaturated fat. If it does no double bonds, then it is a saturated fat. And so that's the difference between those two. When you take glycerol and combine it with fatty acids, you get what's called a triglyceride. So if we break this down in parts, and we just focus over here, we have the glycerol portion, which is the carbons, three carbons, and the OH, OH, OH. And then over here on the right-hand side, we have the carbon chain, the fatty acids, and the carbon chain. So here's a fatty acid and a fatty acid. We have three of them, 1, 2, and 3. When we look at this and we combine the glycerol to the fatty acid, you'll see something that happens that we were first introduced to with carbohydrates, and that if we remove two hydrogens and an oxygen, like this right here, and we do that, it will actually be 3 times, we will make water, and we'll make 3 waters. In addition, we will have an extra oxygen left over. So let me erase these right here. We'll end up with an extra oxygen left over right here, to combine to the carbon that's here. And so we'll actually make an oxygen bridge. Again, that carbon connected here, that carbon connected here with this oxygen. Over here on the right-hand side, we see the same thing. Here's the carboxyl group, except this time we have the chains that are here. These are all saturated chains. And then we can actually see where we take away the water, we'll be left with an oxygen which combines to the carbon. And we would have a double bonded O. And you'll see in just a moment that you still fulfill the bonding rules with carbon by having this double bonded O that is here. Now, because of the double bonded O, the OHs, the oxygens over here, remember that the oxygens tend to pull electrons a little closer to them, thus causing them to have a slight charge. So we consider this portion, because it's charged, to be hydrophilic. Remember that means to water-loving. So this portion right here is hydrophilic, while the portion over here without any hydrogens is hydrophobic, which means that it has no charge. So fatty acids, triglycerides, tend to turn themselves so that the side with the more oxygens turns towards the water, while the hydrophobic end turns away from the water. And if you remember, we kind of talked about this with phospholipids, where we had our head and then our chain, except this was just a two chains, where this portion right here was our hydrophobic end, and then our head right here was our hydrophilic end. So this is the same concept. We actually have several different kinds of fats: saturated, unsaturated, polyunsaturated, trans fats. We can just focus on two kinds. Saturated fats, or lipids, actually come from animals. Again, saturated means that it is saturated with hydrogens or filled with hydrogens. I like to think of it as kind of water, that a sponge filled up with water, that it's saturated with water. No more water can fit into it. And then on the end here, we have the carboxyl group that is here. And remember, this OH is what's removed in order for it to connect to a glycerol. An unsaturated fat comes from plants, nuts, fish, etc. And these are unsaturated, and then they have the double bonds that are here, meaning that they are not filled with the maximum number of hydrogens. And the reason they're not is because they have double bonds. And again, this portion right here is the carboxyl group. So if I didn't make that part clear, it is the OH group and the carboxyl group which combine, that allows the glycerol and the fatty acids to combine. A saturated fat, which are normally not as healthy, you take the fatty acid, you'll see that you have three of them here. The tail end that's here are filled with the carbons, right? And this is the hydrophobic end. And we take the carboxyl side and we combine it with the hydroxyl side of the glycerol. And again, we remove water. And by doing that, we create what's called an oxygen bridge. We do the same thing with the second fatty acid. Here's the carboxyl group, and here's the OH group. Carboxyl group and the OH group. And then we will end up removing three waters, just like we saw earlier, right here with three waters. Here's the two H's and the O. And this is how you make a triglyceride. If you make an unsaturated triglyceride, it's the same concept that we talked about before. This would be where the OHs are on the glycerol, and this would be the carboxylic acid portions would be on the fatty acids. The unsaturated, the only difference is in the tail, where we have the double bonds that are here. So everything else is the same. Everything else is the same. This is our oxygen bridge that is here. And this is a good time to mention that, with fats, they are composed of the same components that form carbohydrates, C, H, and O. And this is what happens to unused carbohydrates. When they're not used within the body, they will turn to fats. And so that's something that you need to consider. And so that's why there's a lot of similarities between with the oxygen bridge, the carbons, hydrogens, and oxygens. That's why you need to consider that when you're eating excessive amounts of carbohydrates. Cholesterol is another form of fats. They're used as part of the cell membrane. We actually talked about that when we were talking about C-GADI actually getting into the cell membrane, getting through that portion. It's able to trick the cell to bring it in. There's the primary function of the cholesterols in the cell membrane is for cell signaling and helping to form other cells. Another form of fat that is actually, was really popular for some time, but has just recently, maybe the last 10 years or so, is found to be really bad for us, is trans fats. And today you actually see a lot of packaging that says, Doritos 0 grams of trans fat. And really what the trans fats were used to do is to increase the shelf life of foods, to increase the texture and the way foods would taste. And it turns out that trans fats are actually man-made fats. And what's done is you're taking the fats and taking like an unsaturated fat, like a vegetable oil, and adding hydrogens to it. And when you do that, you make the oil much more stiff. Like for example, comparing vegetable oil and Crisco. So if you were to compare those two, you would actually see that one's more of a liquid while one's more stiff. Now, the reason why trans fats are really bad for us is that they increase the amount of this, what's called an LDL, low-density lipoprotein. These are bad cholesterols, and they run around into your bloodstream, but too many of them, if you had too many of them, what they do is they build up, and then they start to clog your arteries. Trans fats also will decrease your HDLs, which are high-density lipoproteins. Now, these are the good cholesterols. You may have heard of bad cholesterol and good cholesterol. LDLs are the bad, HDLs are the good. And what the HDLs do is they work around your blood, and what they do is they pick up the LDLs. So if you're a normal person, you have some LDLs in your blood, you're also making HDLs. It's not that big of a deal because your HDLs will take care of your LDLs. If, however, you're eating a diet that has high trans fats, the trans fats is reducing your HDLs and increasing your LDLs. And you can look some more things online about it. It's all pretty interesting stuff. This last one I thought I'd offer some extra credit on. It's something that, if we had more time, and maybe even if I do find that we have some time, it's something I've introduced kids to in terms of an article from Time magazine. It's something called olein, and I'll just pose you this question. With today's technology, can we make a man-made fat, other than trans fat, which will taste great but not build up in the body? And what it turns out that we have had some success with this. And so I'll offer some extra credit if you research olein, or it's called olein or olestra. It's an interesting story behind it. I'll offer some extra credit. I'm not sure how much. I guess it kind of depends on the quality of the work. If you bring in an article, so you have to bring in the article along with a 100 to 200 word conclusion. And you need to be able to answer the following. What is olein? Why was it supposed to be great? And what are the side effects? And then the last one, number 4, is it still used? If so, where? Do you know anyone who has had it? I can tell you that I have had it, and it's interesting that it was something that sold in Michigan not too long ago. That'll wrap it up. The next video will be more about how these things are digested. I just wanted to do them in steps, and so on Wednesday night you should be able to see a video on the digestion of these things, which again will be very short. Thank you.